Hello, welcome to 13 Universal, and we are back to A-level CIE biology. This is Shedro with you. So we are looking at all topics theory paper 2, series 4. We have done 1, 2, 3 for you. Okay, so join us as we move to 4. Get your pen and paper. All right. Says the cells in a tissue are often at different stages of the cell cycle, right? That's the first question there. So look at this tissue, guys. What do you see? Right? So say fig 1.2 shows cells at different stages of the cell cycle. Different stages of the cell cycle, okay? Okay. And let's look at what the question says. Identify the stages of mitosis occurring in the cells labeled B and C in fig 1.1. So guys, before you start solving questions, master your background, the stimulus materials, your text, right? They said the cells in a tissue are often at different stages of the cell cycle, of course, and you can see that this is under stimulus material. So look at these cells. This is anaphase, towards the end of anaphase, beginning of telophase. Now, what do you think that cell A is? Which stage is it? Okay, that looks like anaphase. B, what phase? Now, why do I say anaphase? You can see that the sister chromatids are separating. So B looks like prophase because they are condensing and coiling up. The chromatin or chromosomes are coiling up to become more visible. All right, this see the chromosomes are lined up at the equator, so that looks like metaphase. Okay, all right, this looks like, all right, prophase. This is still interphase. You can see no major activity of chromosomes. It's not actually condensing. Okay, this looks like interphase 2, right? Okay, so fig 1.1 shows cells at different stages of cell cycle. So you can see that this is maybe G2 because chromosomes have been doubled here. So identify the stages of mitosis occurring in the cells labeled B and C. I think we've identified B. B is prophase. Why? Because the chromosomes are coiling up. Can you see they are coiling? They're condensing. C is metaphase. Why? Because chromosomes are lining up at the equator of the pole or the cells, equator of the poles. All right. So let's see what the mass scheme says. It says prophase. All right. It says ignore early, mid, late. Just say prophase because that's not clear. All right. So we have C is metaphase, accept early anaphase because it looks like they're about separating. Okay, this is the mass scheme. All right. So let's go to the next question quickly. So just 2 marks. The cell, the same question. We want to look at the next level question. All right. It's a figure. Okay, we have seen that. All right. It says describe the behavior of the chromosomes in the stage of mitosis shown in A. If you check A, if we go back, all right, to the next slide, of course, that A is anaphase. Okay. So if you go back, you see that A is anaphase. So what happens in anaphase? And anytime they say describe the behavior of the chromosomes in the stage of mitosis shown inside. This A is anaphase, right? We mentioned it. All right. All right. So describe means show us sequence of events. How do they occur? The first to the next and to the ones following based on their priority, how they are occurring. Okay, remember that in anaphase, guys, don't forget, you may try and pause this video and try it. Okay, pause it and try it. So in anaphase, remember that the first thing that happens in anaphase excuse me, the first thing that happens in anaphase is that the centromere splits. Okay, you know what is called centromere, guys. I've seen a chromosome before, right? And of course, these chromosomes are lining at the equator. So there is time to separate, okay? Because metaphase leads to anaphase. So the centromere has to split. Now, this has to split, yes. So that's the first event, centromere splits. Okay, and then the next thing that happens is that at the poles, guys, if you remember, let's assume this is cell. Okay, so at the poles we have spindle fibers. All right, spindle. This is central at the pole. They produce spindles. Okay, these spindles, they are attached to this, right? They are attached to it. Let's do it well. Okay, yeah. So what is going on, guys? So you see the spindles attached, all right, to the connector core of this sister chromatid. Remember that each of these arm of chromosome is called a sister chromatid, guys. All right, this is sister chromatid. Okay, so this is centromere. Centromere joins them. It joins the sister chromatids because they are genetically identical. So at the pole here, right, if you follow the sequence, what happens is that the microtubules shorten. So when microtubules shorten, what happens? That's the next one. So microtubules shorten, and when they shorten, so remember the first event we are describing, guys. We are describing. Describe means tell us how. The prose format answers the question how. Sister centromere splits into two. The at the poles, the microtubules or spindle begin to shorten or undergo depolymerization, and when they shorten, then the spindle, okay, contract, pulling sister chromatids. So sister chromatids are pulled to the opposite poles, right? Oh yeah, they are put to the opposite poles by the centromere, with the centromere leading. So if you look at this part of my drawing, so they are pulled to the opposite pole with the centromere. Centromere is always leading. Okay, so let's assume that this is the first. Okay, this is it. This is sister chromatid lagging behind. Right, centromere is leading. Now this is spindle. Just want to show you how this is happening. Okay, this is centromere attached, this sister chromatids. Okay, centromere is the one leading. Okay, now that's how what happens. Okay, all right, this splits, this shorten and contract, pulls the sister chromatids apart to the opposite poles. Okay, so what do you think? Let's see what the mass scheme says. So mass scheme says any three from identical chromatids for sister chromatids, right? Let me clean up. So sister chromatids, okay, don't worry. You can see it, sister chromatids assume point related to anaphase unless stated as telophase. All right, so anaphase, sister chromatids separate. So they separate there. I accept idea of pulling sister chromatids apart. Let me clean this, okay? Clean it up so you can see it very well. All right. So we can clean up. Yeah, you can see that. So guys, look at this. At the next one, let's look at the next point there. Yeah, idea of spindles pulled apart. Like I said, they are pulled apart. Next one, metaphase, okay, start of anaphase, movement of total chromosomes, you know, to the opposite poles. That's what happens. Reject contents of some chromosomes of two chromatids. Okay. See what is separating in mitosis is no chromosome, is sister chromatid. All right, so that's why it's reject if contains movement of sister chromatids to same pole. They move to different poles. Sister chromatids move to the opposite poles. They are pulled apart to opposite pole, or a movement to opposite poles by spindle, just like what was said. See that centromere divide to start. All right, so this gives us the bullet point that should be there. You can see that leading to, but you know, you must follow, right, the sequence. Okay, so you can pause the video and look at it very well. But if somebody mentions telophase, you say that they are decondensing at telophase. So let's go to the next question. Telomerase is an enzyme that is active during interphase in some cells. Telomerase helps to maintain telomeres present on chromosomes. Okay, they help to maintain telomeres. Telomeres, where are they found? Okay, it is a chromosome. Okay, so where are part of my diagram? Okay, this is sister chromatid, this is another sister. So where are telomeres found? They are found at the tip, okay, at the end of chromosome. So they are repeated repetitive base sequences that prevent the loss of genes. Okay, so let's go. So state the location of telomeres on chromosomes. So at the ends of chromosomes, all right, look at it. At both ends of, okay, of interface chromosomes, if you like, but both accept on the ends of a chromosome. Okay, all right, interface, because cells are about to start dividing. Okay, bodies is found at the end there. You can see that. All right, quickly, let's go to the next question. Just one mark. Some people with a long lifespan have cells showing a higher than normal activity of telomeres. Telomeres, okay? Telomerase is an enzyme, all right, that replenishes telomeres. So some people that have long lifespan have cells showing a higher than normal activity of telomeres. So why do people live longer? There's an explanation to that. Telomeres explains that. Telomeres also explains why people age. The shorter your telomeres, that means that the less the number of division, the person is going to have short lifespan. But if somebody has longer telomeres, they will have longer lifespan. So telomeres help to determine lifespan. Okay, so just why a long lifespan could result from higher telomeres. Now, why would a long lifespan, all right, determined by higher telomerase activity? Remember that telomerase help to maintain, all right, or replenish telomeres. It replenishes telomeres, so it maintains the size of telomeres. And once it maintains it, okay. Once it maintains the size of telomeres, that means that the cell can continue to divide. So have increased cell cycle, or DNA replicates more. Okay, and then because of that, we have more number of cell cycles, because remember that one of the roles of telomeres is to, all right, ensure that cells continue to divide or DNA replicate continuously. All right, so every division telomeres deplete, and that's why we have apoptosis. But if you have telomerase enzyme, so the telomerase enzymes make more telomeres, so cells continue to divide. All right, and then we have more number of cells, and we have less genes lost, or no genetic information lost. Okay, there's no apoptosis. Okay, the cellular activities, all right, increase. And if you have more cells formed, that means that if there is any damaged cell, it can be replaced, so the person looks a bit younger. All right, because damaged cells are replaced. Okay, tissues are repaired. So don't forget, it must be a logical, because this is just why. The reason is that telomerase maintains telomeres, so more cell cycle, more DNA replication, more cells are dividing, and we have more, of course, damaged cells have been replaced, and then genes are not lost. Okay, and then of course, apoptosis does not occur. Apoptosis is programmed cell death, right? But look at what Maskim says. So it's the telomeres are maintained for longer length of time. Why? Because this actually makes telomeres. Okay, so that allows increased number of cell cycles, DNA replication occurs. So these are the reasons. So it allows mitosis or cell division to occur. That accept, A means accept, reject, speed up. Well, it doesn't increase the speed, it just increases more number, not the speed. Okay, prevent loss of genes. So of course we have more telomeres to prevent loss of genes. Now the root receives, assimilate to store, so it carries out its function. Less genes are lost, ignore loss of genetic material. So with genes, all right, greater ability to replace damaged worn out cells, ideal metabolism remains optimal. So these are the things Matt Kim says. Let's go to the next one. The number of cases, this is the second question. Number of cases of tuberculosis TB is affected by many biological, social, and economic factors. Okay, so biological factors like high rate of mutation, and that's antigenic variation. It mutates fast. We have antigenic concealment. All right, it's an endoparasite, so it lives inside the host cell, dormant for years. Social factor, people don't have access to information about transmission cycle. Okay, ignorant migration, civil unrest could cause that. Economic factors, people live in overcrowded environment, poor living conditions, okay, lack of ventilation, all of that. So let's look at what this says. Please, I want us to look at this. So this is a graph, and you will always answer graph questions. So if you 2.1, then the number of reported cases of TB in the USA between 1980 and 2000 is shown in fig 2.1. That's it here. 1980 to 2000. All right. Please, this is reported cases. Remember that whatever that is on the x-axis is the independent variable. So this is affecting this. So this is what we're measuring, number of reported cases. That's the dependent variable. There's no way. They will always ask you to describe this graph. Guys, now I want you to pause this video and describe this graph. This is data analytics. Show us trends and patterns and relationships. The relationship between year here and number of reported cases, these two variables. All right, show us. Now, if anytime they're going to ask you that, to describe patterns, guys, the first rule to describing patterns, don't forget it, is that you must be able to identify the general, all right, trend. Can you see general trend? The general trend is that this is decreasing, even though it decreases, increases again. All right, it decreases, but generally from 1980 to 2000, all right, the number of reported cases declines or decreases. That's a general pattern. Okay, now you can see that if you look at specific features of this graph. All right, now, but once you state a pattern, you must quote data to support it. For example, you can say that, and for you to that, look at this number of cases. All right, you must know the value of one box. For example, from here to here is 5 boxes counted, and that's 2000. Right, so one box will be 2000 divided by 5. That's 400. So you see, to support that is that in 1980, all right. Can you see this box here? That's about, remove 400 from 28,000. That's 2007, because that's one box below. That's 27,600 in 1980. All right. We'll look at it in 2000. It has decreased from 27,600 to what? So I'm quoting data to support the trend. Okay. To about, that is 16,000 plus 400. That's 16,400. That decreased from 27,600 to 16,400. Right. So you now look at specific features. Okay. For example, from 1980 to 90, if you check this part, this is 5. Okay, 10 boxes represent 5 units. So one box will be 0.5. Okay, so this here is 2 boxes backward. That's 1984. From 1980 to 1984, that's a sharp decrease. Okay, that's 1980, 81 to 1920, that's a sharp decrease. Right. All right. So it mentioned specific features of the slope. From 90, this is 1992, okay, and half. All right, 6 months. You see what is happening? It decreases continuously to 2000. All right. So you know, these are, just look at this. So let's go to the next question. It says, describe the trend. I said it. Describe the trend shown in fig 2.1 between 1980. So look at, let's go into the, it says maximum if 2 number reported cases. So you have to state quote data, any 3 from. So that's 3-mark question. Overall decrease in number of reported cases, all right, from 1980s overall over the time period. So what is happening? This is data analytics. So it decreases. The number of reported cases decreases from 2,600, all right, to 16,300 and 400 cases. So you quote data. There's a decline. All right. So once you state the trend, you quote data to support it, guys. That's how it works. Additional trends, okay, decrease from 1980 to 1984. You see that I showed you decrease after 1982. So all those parts where decreasing, you have to mention them. Little change, slight increase and decrease, all right, on the graph. Use comparative value, all right, to support your point. Quote data, all right. That's data analytics. You must state the trend and you must quote data to support it, identify specific features. Now the next question says, the number of cases of TB between 1984 and 1992 in the USA may have been affected by an increase in the number of people infected with HIV. See, HIV, right? An increase. HIV is an increase at this point, right? And you know, increase in tuberculosis, HIV, all right, cases also always lead to increase in tuberculosis cases, and that's what we want to know. They say explain how, explain, give us reason, or show us how an increase in the number of people infected with HIV could have affected the number of tuberculosis cases. Okay, so how does HIV affect this? There's a correlation between HIV and TB cases. There's a correlation, and it's a positive correlation, that as the number of cases of HIV increases, the number of TB cases increases. It may not be a causal, but there's a correlation. It may not be a causal relationship, but there's a correlation between them. Why? Why does that happen? Guys, don't forget that HIV, all right, AIDS, weakens immune system. I'm explaining how. It weakens immune system. Why? Because it reduces the number of T lymphocytes or T helpers. Right? The target site of the pathogen is the T helper cells. All right. So once T helper cells are affected, all right, less B lymphocytes will be stimulated. Okay, so it affects also humoral immune response, which is the immune response that produces antibodies. So less antibodies will be produced. Okay, so immune system is weakened, and once it's weakened, TB is an opportunistic it's an opportunistic infection. All right, opportunistic infections are infections that occur only when the immune system is compromised or impaired or weakened, guys. Right? So that is it. So once the immune system is weakened, we have less activity of immune cells, less cellular and humoral immune response, less antibodies produced, and TB, dormant TB becomes activated. All right. So how do you say it? Well, this is because HIV weakens the immune system, right? And TB is an opportunistic infection. It happens when immune system is weakened. All right. And how does it weaken immune system? HIV, all right, reduces the number of T helper cells, and T helper cells stimulates B lymphocytes. So that there'll be reduction in B lymphocyte activity. All right, less antibodies made, and all of that. Let's see what my scheme says here, guys. HIV infected people may develop, they may develop a weakened immune system. People with people with HIV have it, okay, like the same way of saying it, or poor immune system. Idea of more susceptible to disease. So people that have HIV are susceptible to this pathogen. All right. Why? Because TB is an opportunistic disease. Okay, this is why increased risk of latent TB becoming active in many people living with HIV. Many people living with HIV, they're at higher risk of having TB. You can talk about detail of weak immune system, decrease in the number, of T lymphocyte. That's it. T helper cells, these cells, okay, reduce the number. And then what's the impact? Poor humoral, because T helper stimulates appropriate B lymphocyte, remember, to divide and then of course differentiate to plasma cells and memory cells, remember. All right, that's it, guys. Idea of more cases of TB owing to more people infected. So if more people are infected with HIV, all right, so more people with HIV increase risk of spreading to uninfected people. So more TB cases to be spread. Mycobacterium tuberculosis is a species of bacterium that causes TB. Strains of M. tuberculosis have evolved resistant to some of the antibiotics used to treat disease. State two ways that the impact of antibiotic resistance can be reduced. So how do we reduce the impact of antibiotic resistance? How do we reduce it? Okay, now antibiotic resistance is when an antibiotic loses its effectiveness in the treatment of a particular infection or bacterial infection. Right? Antibiotics are used to treat bacterial infection when it loses its effectiveness. All right. So maybe of so many factors. All right. So how do we reduce it? How we reduce it, number one, is that antibiotics must be prescribed across the counter before they are taken. Okay, people should not use leftover antibiotics, and of course, people should complete the full course of treatment. In tuberculosis, for example, the treatment time is usually minimum, I think 6 months, 9 months, So there's a chance that people stop after some time. So we use what is called DOTS, direct observation therapy, observation therapy scheme, when people are watched every day to take their dosage. Okay, so dosage must be completed, right? And very importantly, guys, don't forget that we can make more antibiotics, okay, to reduce the impact. And we should also practice more hygienes in hospitals. For example, use more antibacterial and antimicrobials, all right, to reduce the load of antibiotics. Pathogens, all right, around hospitals, because people go to hospitals with loads. So how do I answer this? Just a state two. So number one, okay, people should complete dose of complete complete course of treatment, right? Number two, people should use antibiotics when necessary. Antibiotics should be used when necessary. Number three, antibiotics should not be used on animals, right? Human antibiotics should not be used to treat animal infection, because that could lead to that. Okay, all right. So another point there, like I've mentioned, that um, the appropriate antibiotics should be used, the correct dosage, the right antibiotics should be used. For example, if you are treating infection that has to do with broad spectrum, you use broad spectrum antibiotics. Okay, look at masking. Any two. Prescribe, take antibiotics only when absolutely necessary. So please don't forget this. Yes, do not use it for viral infections. It's not a preventive medicine. Make sure, all right, make sure correct, effective antibiotic prescribed, used. All of this was said, but you memorize it because biology repeats questions. Make sure people take the correct dose. Yeah, complete course of following instruction. Develop new antibiotics, right? Use other antibacterials. Okay, that's it. Treatments use different. Yeah, you can combine two different antibiotics. That's synergistic effect. Mentioned, prevent infection, break transmission cycle, reduce need of antibiotics. You can use vaccine, hygiene in hospitals, quarantine people who have it, report cases. All right, reduce control use of antibiotics in animals, animal use for food. Okay, educational, professional, all of that, guys. Monitor it. So that is it, guys. Let's go to the next question. TB can affect every part of the human gas exchange system, including trachea, bronchi, and alveoli. The cartilage in the bronchi can be damaged. Some people with the cartilage in the bronchi can be damaged in some people with TB. Suggests the effect that damage to cartilage may have on bronchi. So the question is, what is the role of cartilage? Cartilage provides support. So if you damage it, there will be loss of support. Okay, if you damage cartilage, then the bronchi will collapse. Okay, that's it. All right, that's just one mark. Table 1 point shows 4 types of cells, 3 structures found in the gas exchange system. Complete table 2.1 to show the distribution of cells, cell types in each structure of the gas exchange system. Okay, before I show you the table, guys, I want to ask you, so use a tick if the cell type is present in the structure, and a cross if the cell type is not present. Put a tick or a cross in every box. Okay, it's been done for you. You don't ask you a question. Write it down. In which part you have trachea, bronchi, bronchioles, and alveoli? Which of them contains ciliated epithelium? Okay, ciliated epithelium is found in trachea, bronchi, and bronchioles. It's not found in alveolus. Which of them contains goblet cells? Of course, goblet cells found in trachea and bronchi. Which of them contains smooth muscles? Trachea, bronchi, and smooth muscle. Which of them contains squamous epithelium? It's just alveolus. Okay, so it's been done for you. So look at it, study it, don't forget it. So goblet cells, ciliated epithelium, smooth, they're not found in alveolus. Okay, squamous epithelial cells are found in alveolus. All right, so you can see bronchi, bronchus, and trachea, they don't have squamous epithelium. All right, but it has related epithelial cells, gallbladder cells. All right, this is where we stop for this. Right, so we're going to be bringing series 5 or series the next is yes, series 5. Yeah, thank you. Share this video, send us, make comments, tell us what you need. All right, if there's any area we have not touched, let us know. See you. Bye. Next class. Okay, bye.